Welcome, everyone. In today's module, we're going to be talking about the Chemical Characterization test series governed by ISO 10993-18. Chemical characterization is a great place to start for any medical device company looking to understand the biocompatibility and risk profile of their medical device. It's very often the very first place you start, and the results that you get from chemical characterization are very often going to justify either the necessity to do more robust studies or potentially open a window in justifying very few. For many 510K devices with well established risk profiles, this might be one of the very first and few tests that a medical device company has to do. Whereas perhaps with more risky PMA devices, class 3 devices, very often you have to do this, and then that'll open the door and demonstrate the risk and necessity to do many other more complex biocompatibility tests that the FDA certainly recommends or almost requires that you do. So let's talk a little bit more about it and what it is. Now, chemical characterization is the process of obtaining chemical information accomplished either by information gathering or by information generation through testing and perhaps literature reviews or certain benchtop tests. And it revolves around the primary concern of the chemical profile from a medical device that can leach out into the body. I've said it before, no one cares what's inside the medical device unless it has the potential to get out into the body. Again, my exaggerated hyperbolic example is that you can have a medical device made out of arsenic, maybe not be a good idea, but the arsenic is only an issue if it solubilizes and gets into the local tissue systemically within the body. It's only the solubilized compounds that can wreak havoc on our body and produce some sort of toxicological or pharmacological effect. So therefore, the FDA only cares about what comes out of the material. Now, very often this can be governed by what's in the material in the first place, so maybe they wouldn't accept an arsenic medical device. But still, nonetheless, the concern is what gets out into the body. Now, the two key words that we use to describe these, and very often you hear this as E&L testing for extractables and leachables. Now, leachables is really what's going to, in a real world condition, get into the body. And it's going to be the focus of what the patient might be exposed to under the intended use of the product. Now, as I'll talk about in some of the next few slides, that might not be practical to test. So we have to almost forcibly extract chemicals under exaggerated laboratory conditions in order to approximate or see what the worst case scenario would be under those non achievable and perhaps unpractical real world conditions to replicate in a lab. So, very often I hear these two terms used synonymously. Just understand that leachables is what is the real world condition, and extractables it even has that sort of connotation of it's being forced. Someone has to extract something from the medical device, and that's exactly what we do in benchtop testing. So to expand on that idea a little bit further, again, leachables is what comes out under practical real-world conditions. And one of the best examples I have to describe that is a medical bag, like a saline bag. So this would actually be a medical device, a product that comes with saline, sterilized, already ready to be treated to the patient. That saline has been sitting in that bag for however long the shelf life is of the product. And literally, what the patient receives is what gets leached out from that bag, plasticizers, parts of the manufacturing process that were originally part of the bag. They make their way into the saline, and what the patient receives is literally 100% of the items that leached out. And it's the best representation of a one-to-one dose to receiving chemical profile of what the patient gets. In order to do leachables testing and chemical characterization, you just take a sample of that saline and you look at the chemical profile, and you can be confident in saying, well, this is exactly what the patient is going to get. Now, in other conditions, so let's say like a knee implant here, you can't replicate that. If you have a knee implant out of the box, it might be sterile, but you have no way of understanding what the dose of the chemical profile is going to be into the patient. You can't somehow test that, as in put this into a patient and in real time monitor what's being leached out of it. You have to, under exaggerated conditions, somehow replicate that in a benchtop scenario and put in some sort of, saline, a synovial fluid replicant, a physiological solution, and try and estimate what comes out of it. And you can't do it at over a 10-year lifespan of however long it's going to be implanted into the patient. You have to force it. You have to exaggerate and make sure that you can sort of approximate what comes out in the lifetime of a patient in a very exaggerated and accelerated short-term profile in the bench, which is why we have to extract something like this because we don't exactly know what the true real-world profile is going to be. We have to force it, and therefore we do an extraction study. So those two types of medical devices are the perfect sort of opposite ends of the spectrum, ways that we have to estimate or know exactly what's going to get in touch with the patient. Now, we want to understand what the chemical profile of that extraction or leachable test is. Well, how can this help us? There's a few different applications of use that a medical device company might apply this towards. So, the primary one is to support the biological safety of their medical device and the risk assessment profile as outlined in the Dash 1 standard. The next step after doing chemical characterization, no one looks at chemical characterization by itself. You use that to support what's called a toxicological risk assessment. And we'll talk about this in the second half of the lecture, which is governed by the Dash 17 module. And this is really where you understand what's the downside, what's the true risk of any of the chemicals that I observed in my benchtop testing. It's also going to be used to support potential materials to incorporate the screening of new materials into medical devices. So this very often goes hand in hand when you need to show equivalence to a new version of a medical device. It might be you change your manufacturing method and you have to, change suppliers of a particular material and you need to see how one material compared to a prior one. You need to see how it compares to predicate devices. You need to look into further prototypes and understand how your assembly method, again, your manufacturing method, how it's going to affect any potential changes in your medical device. So very often as you change or tinker or optimize a medical device, you might relook into chemical characterization to understand how any of the changes you implemented change the overall chemical profile and therefore the toxicological risk or TRA profile of your medical device. Now, medical devices are oftentimes very complex, containing of multiple complex parts to them. Certainly, the more you get into surgical robots and the like, the more complex it's going to be. Even something as simple as, on the opposite end of the spectrum might be a scalpel or something that might be just a single piece. And really, whenever we look at the chemical characterization or the chemical or extractable and leachables profile of a medical device, the only thing that the FDA cares about is what comes into direct or indirect contact with the patient. Now, direct contact means that whatever part of that medical device is actually literally going to be implanted into the patient or somehow going to be received by the patient, like the saline part of a saline bag. Now, the indirect parts are the things that actually don't make it into the patient, but somehow had contact with those that have direct contact. So, for instance, the saline bag that does not get implanted to a patient has indirect contact with the patient because the saline has been in contact for a long time in that bag before it's actually delivered to the patient. So that is, of course, a concern to the FDA. Now, here in this sort of pre-filled hydrogel syringe of a novel spinal prosthetic, we see here a number of different assembly parts where that pre-filled syringe has the hydrogel that's going to have direct contact with the patient. The syringe itself has indirect contact. It never gets implanted into the patient, yet it contacts the gel. So the FDA is very much concerned about the chemical profile and the extractables and leachables from that syringe. And same thing, anything that's in line with the implantation cannula, the tubing, the pressure gauge, anything that comes into indirect contact with that hydrogel as it's being delivered. Whereas any of the other plastic parts, the metal parts that make up this delivery device that never touch the hydrogel ever are going to have no toxicological consequence to the patient and therefore have no effect on the chemical characterization profile of the medical device. Now, the broad steps in E and L testing, extractables and leachables testing, includes the bulk part of chemical characterization, which can be described in three discrete steps. The first part is the extraction step itself. This is where you actually get all the chemicals and forcibly extract them out so you have them in some sort of testable solution. From there, we go to the detection step where we use a variety and a battery of benchtop analytical techniques in order to understand what chemicals or molecules are in that extracted media. And then once we detect those, then we identify exactly what peaks and we actually label the compounds that are in there. And you'll quantify the amount that are in there as well. So then you know exactly what chemicals came off and in what sort of concentration or magnitude. Now, as I mentioned before, Chemical characterization isn't really just done by itself in a vacuum. It most often inevitably comes down to identifying the risk associated with those molecules, which is step known as the Toxicological Risk Assessment or TRA. And that's governed by a totally separate ISO subchapter, the 10993-17 subchapter. And we'll talk about that a little bit more. But these two ISO standards often go hand in hand in order to really fully characterize not just the chemical profile, but the full risk profile of your medical device. Now, let's jump into the actual extraction part. Let's understand what scientists actually do from a benchtop level in order to carry out this series of testing. So, extraction involves a number of different solvents where, again, you want to, under exaggerated conditions, not just use a physiological solution, but use a variety of solvents that could possibly extract anything and everything to represent the worst case scenario. So the standard actually calls for that long-term implants use a minimum of 2 extraction solvents, and these have to be of different polarity. So you might have an organic and a non-organic solvent in order to try and extract everything out of there. Now, the FDA actually recommends to go a little bit above and beyond this to use 3 solvents that kind of run the gamut from polar, non-polar to semi-polar for long-term implants, unless you can justify that. So usually, unless you've done this enough and you can somehow have enough data to back up that for your particular medical device and your scenario, you don't require 3 extraction solvents, then you usually have to do 3 in order to meet that recommendation. Now, all extraction runs need to be done in triplicate. So that means for one chosen solvent, you need to have at least 3 different replicates in order to sort of replicate that process. Again, unless you have low variability benchtop testing that can support some sort of justification for not doing so. Now, then later, when you actually do the analytical runs, each individual replicate typically needs to be analyzed in triplicate. So that means that one vessel or one of 3 of your polar solvents, you need to run that single vessel, that extraction solvent, 3 times on the same identical solvent in order to understand what the variability is of your actual detection system. Now, then from there, you might only report the maximum, aka the worst possible scenario of those 3, but as you can see, these numbers start adding up pretty quickly. If you have 3 solvents from which you're doing 3 extractions and then 3 analytical runs or 3 replicates of those, then that's going to be a total of 27 runs or samples per method that you're running. So already this is starting to get pretty large and pretty comprehensive. Now that's almost as little on the extraction part and the sample prep that the dash 18 standard covers. The reason why is that there's actually a whole entire other standard that just simply talks about the sample preparation and the preparation to how to do these steps in the first place. And that's governed by the dash 12 standard that talks about sample preparation and reference materials. So we'll actually switch over to this and talk about this for a little bit in order to understand a little bit more how we're going to actually run these samples. So within the dash 12 standard, they talk about the solvents or replicants that you might want to do. And they give tables on the recommended polar, semi-polar, and non-polar ones. And they talk about their various polarity indices, the boiling points. These can be helpful for a number of different extraction runs. For instance, many different analytical benchtop methods are really suitable for polar compounds, but not very much for non-polar compounds. Or some of them, like gas chromatography, need a compound that's pretty volatile and aren't going to be compatible with something that doesn't boil off or turn into gaseous form very easily. So, very often, analytical scientists will both want to optimize the extraction profile through their chemical of choice, but then also think ahead to their analytical methods in order to make their lives a little bit easier. So, they'll look at these tables, and as long as it's recommended and it's on there, they'll look at some of the solvent properties and then pick from there to optimize their analytical runs. Now, once your solvents have been picked, you need to consider the time and the temperature over which you do these extractions. So the Dash 12 standard gives a table to refer to that can describe both the temperature, their tolerances, and the time that you have to run these extractions through. Anywhere from the minimum contacting materials to what's recommended for the lifetime or long contacting materials. Now, in general, more extraction is going to occur at elevated temperatures and when done over longer periods of time. So, if you have a device that only has minimal contact with a patient, maybe you can justify just doing body temperature for 24 hours to perhaps exaggerate a condition where a catheter might only be in a patient for 20-minute infusion or something. However, you might then have to go to longer times, such as 3 days or 72 hours at maybe body temperature, or if you need to exaggerate that and get the most and get the real world worst possible scenario, you have to go to 50 or 70 degrees. Maybe even for those that have to receive a steam sterilization within some sort of container, they might have to go up to 121°C, which is a sterilization temperature. Now, long-term implantables generally require for the 72-hour extraction. That's the longest that you see on this list. However, you need to consider your own medical device needs. A lot of medical devices might have negative impact through higher temperatures. For instance, higher temperatures can increase polymerization that occurs and reduce the amount of extractable monomers that might have their own inherent risk. Or they might also increase degradation products that, while might be dangerous in themselves, just don't have a lot of clinical relevance. So it's up to the medical device company to pick something that's adequately represent representative of a worst case scenario, but also doesn't somehow go beyond a practical or a feasible, clinically relevant limit within their medical device. Now, those extractions over 72 hours isn't the end of the particular test. So usually the regulatory body will require what's called exhaustive extraction, where you as a medical device company need to demonstrate that all possible extracts have been pulled out and further extractions are just going to produce diminishing and further diminishing returns. So usually the way that you demonstrate this is that you do one of these runs that occurs for 3 days or 72 hours. And you also do that in a parallel sample that you're going to purposely I'll say destroy, and I'll describe what that means in a little bit. But you're not going to use this for that parallel run for extraction for analytical purposes. You just want to see how much total things are in there. And then what you're going to do is you're going to dry that solvent to see how much residue you have in that particular jar. It's what's called a non-volatile residue, and it's the dry stuff that remains in that jar. If you extract a lot of things, you're going to have a lot of salts, a lot of plastics, a lot of monomers or different polymers that remain in that jar or that vessel that you did the extraction. And you'll be able to sort of just see on a scale how much came off of that medical device. So you weigh that material, and then with that same medical device that you had in there, you transfer that to a new jar, you add new solvent, and you repeat the process. And if you're going to do that again, and you normally would find less things get extracted the second time, less things get extracted the third time. Just like if I want a good cup of coffee, I run my initial extraction and I get a great cup. If I wanted to reuse those grounds and I put hot water through it, yeah, I would get, muddy, dark looking liquid, but it would taste crappy. I wouldn't get nearly the same amount of good things as I did in the first, but things are coming out of it nonetheless. So you kind of repeat that process in order to understand, well, what's the maximum that I can get out of my coffee grounds in this analogy or in this exhaustive extraction? You weigh that vessel and you see how much residue, that NVR, did I get that second round? How much did I get that third round? And whenever you get a run whose extraction percentage, whose mass of NVR is less than 10% of the first round, you've demonstrated that you've reached exhaustion. And in my experience, I've had devices where this happened over 7 runs done in series. So that means 7 times 72 hours worth of extractions, and I have 7 different vessels of liquids that I need to analyze separately in order to understand the cumulative amount of things that have come off of that. So again, if you think about that, 3 solvents, 3 extractions, replicates, 3 analytical runs, times however many of these iterative extractions and series you had to do, you're getting higher and higher multiples of that original number. Another thing that has to be considered whenever doing these extractions is what volume you're actually doing this as extraction in. If you can imagine, if later we actually need to detect what these compounds are that are coming off, if I were to put a scalpel or a knee implant into a 10-gallon tank of ethanol, I might run that solvent during the after the extraction run through an analytical instrument, and lo and behold, not find anything because I had such a dilute pool that I used that it's not feasible for even a highly sensitive instrument to be able to detect anything. So therefore, there's rules that govern how much or how little volume you should use whenever doing these extractions in order to have an expectation that you should be able to detect something of consequence in there. So again, the Dash 12 standard has a nice table that uncovers this and goes through, depending on what the material size, the thickness, the a mass of your device, whatever it might fit through, and let me give you some examples in the third column here. Will define the extraction ratio, usually in typical of the surface area, sometimes the mass to volume. Surface area is the preferred choice because the governing surface area is what's going to define how much can actually get extracted out, right? Everything has to go through the surface of a material in order to be extracted or leached out. If you have a smaller surface area, the better. If you have a really highly porous a medical device that has a really high surface area because that high porosity, then you're going to have a lot more exposure. And that's going to govern exactly how to do this extraction ratio then. Sometimes you have irregularly sized medical devices that don't really have a true thickness, in which case you might have to do it with respect to mass. So now that we've covered the extraction phase, let's talk about the detection phase. And I really like this passage in the ISO standard on the analytical methods that describes the detection procedures that says a single method cannot be qualified for all potential analytes, and it is not possible that a single method produces highly accurate, precise concentration estimates for all potential analytes. Even if you're not an analytical scientist, I think it's somewhat intuitive to understand that there's no one size fits all method that is best for detecting all things. Even the most sophisticated instruments might only be best for detecting volatile compounds, but not non volatile compounds, and vice versa. There's other elements that are only good at detecting metallic or elemental substances, whereas others are best suited to look at large, complex organic molecules. So it inevitably requires a battery of tests in order to understand the full profile. And luckily, the ISO standard gives you a nice breakdown of some recommended, maybe expected sort of methods to use. And that kind of ranges usually based on the volatility or the organic versus non organic nature of whatever products you're trying to get. And usually you want to cover all of these bases. So, for instance, for volatile organics, you might look at to headspace gas chromatography. Whereas non-volatile organics might look at a liquid chromatography method with some sort of mass spec sort of detection method. Elementals, you typically look at ICP mass spec. And then there might be ions or cations where you might look at ion chromatography. In general, my experience, the primary tools for looking at organic extractables are going to be gas chromatography and liquid chromatography paired with a mass spectrometer. And then for elementals, you're going to look at ICP mass spec, which is a great, fantastic tool. Now, within each analytical method, a common analytical question is to understand what's our threshold of detection? What should we aim for? What kind of sensitivity do we need? AKA, how low do we go in our detection sensitivity? It's understood that some compounds at low concentrations can be healthy, whereas at higher concentrations, they can be unhealthy and toxic to us and have consequences to us. So therefore, the FDA and other regulatory bodies need to look for some sort of minimum threshold of a detection capability in order to understand, are you even capable of detecting those potentially harmful amounts? It's very possible that any analytical method could say, hey, we looked for arsenic and we didn't find it. And is that because you just had a poor sensitivity in your instrument? Or is that because you had a great sensitivity and you can say with confidence that arsenic is not present at levels above a picogram per milliliter or something like that? So let's describe how some of these places describe these minimum thresholds. Now, the most common way of describing this required threshold is using what's called the Analytical Evaluation Threshold. It's a threshold below which the analyte need not to be identified or qualify leachables or extractables or report them for potential toxicological assessment, meaning that anything below this is not of the concern of a regulatory body. Anything above it, though, is something that you need to discuss and put into context somehow. So let's kind of dive into this, understand how it's used. Now, it's really only used for mass spectrometry methods. Methods like ICP, ion coupled plasma mass spectrometry, is so sensitive, as generally acknowledged, it's going to be able to detect things at incredibly low concentrations. So there's really not a concern that you're meeting a minimum. It's for other instruments that are more susceptible to this type of issue, which is why it only comes in the space of those organic compounds that look at gas chromatography or liquid chromatography coupled with mass spec. So, a core component of the AET or analytical evaluation threshold is going to be some sort of rule of thumb that describes a risk based toxic exposure per day to a patient. So, for instance, the dose-based threshold is that sort of a chosen threshold. We're going to talk about that on the next slide, but it's expressed in terms of micrograms per day. So, how much can a human survive? An exposure to arsenic. Well, if their food contained, X micrograms per day, they should not exceed this sort of minimum amount. And we'll talk about how they kind of come up with those sort of rules of thumbs. But this is sort of a placeholder for anything and everything that kind of represents a minimum threshold of any and all toxins, not any specific one toxin. Now, that DBT needs to somehow be converted into a concentration in terms of a microgram per milliliter that's applicable to any of our E and L testing, our extractables and leachables. So it's going to be a product of how many number of devices are actually extracted in this sort of media. So if you have a knee implant and you put a full knee implant in a jar, that number would be one. If you could somehow fit that knee implant, multiple knee implants into that jar, and that is a higher number at 3 or 4, that might be required. No reason why you could even do half of a device, as long as you kind of justify that half is somehow fully representative of the entire medical device. So it doesn't even need to be whole numbers. I've done the E&L testing where I've, done fractions, 1.8 medical devices in my numerator there. The extraction volume, that's going to dilute your sensitivity. So, just like in the example I gave previously, if you extract your medical device in a 10-gallon tank, then maybe that's appropriate. Maybe for whatever reason, that's what you need to do. But that's going to dilute your AET and require much higher sensitivity. Again, because it's going to be very feasible that if I have some toxic compound, but I put it in a 10-gallon extraction pool, I'm not going to be able to see it. Why? Because it's not feasible for me to have that level of sensitivity. If I truly want to put it in something of that high amount of solvent, I need to then justify that I have the sensitivity to overcome that dilution factor. Uh, Now, moving on, the number of devices in contact with the body. Again, I might test 3 knee implants at a given time, whereas I might only put one or maybe at best 2 knee implants into the body. So I can increase the number for A in order to decrease my required sensitivity, and then have a lower number for the actual labeled use, how many of these devices would actually be put into the body as a maximum amount. Now that there might be times for my analytical method that I need to dilute my material, it might actually be too concentrated for my certain method, or for whatever reason, I have to do some sort of post extraction processing step where I have to dilute it. That's where that's going to come into play. So that could either be a number less than one or greater than one, if I concentrate it or if I dilute it. Now, an uncertainty factor is a number that's related to my analytical method that sort of comes from our understanding of the variance that's built into some of these things. You don't need to know that off the top of your head. The ISO standard gives some typical uncertainty factors related to those. But before I talk on that, let's look at the dose-based thresholds and understand how analytical scientists and toxicologists came up with one sort of number that represents one size fits all placeholder for all toxic compounds. So the DBT, the dose-based threshold, is based on an appropriate threshold of toxicological concern, the TTC, which really depends on the frequency and duration of exposure. Now, DBTs are generalized values, and it can be used as a placeholder, but the Dash 18 standard does make exceptions for some highly toxic substances that are excluded for this. And this might be cancer causing compounds for which you can't just use this rule of thumb. You might need to actually detect lower based on these particular concerns. But for the most part, the TTC is governed by the exposure within for the medical device. And if you really want to dive down the rabbit hole of this chemical characterization profile, again, we started with the Dash 18. We have to go down the Dash 12 standard to understand the sample preparation. Dash 18 governs the toxicological risk assessment. ISO standard 21726 is what governs the TTC, the threshold toxicological concern, and goes into a higher detail about that. But this table kind of gives those TTC values in order to understand that a bit more. As you can see from the table, those materials that are in a shorter contact with the body, less than 24 hours or from 1 day to 30 days, you might have a much more higher tolerable intake, which again reduces the sensitivity required because this number will be used in the numerator. You don't need as sensitive an analytical equipment for something that's only in a short duration contact in the body. So that 120 micrograms per day is a much higher threshold that you need to meet. And when I say higher threshold, that means lower sensitivity. So that's more favorable for the analytical chemist. If you're going for something that's a longer exposure, greater than 30 days, greater than a year, greater than 10 years, then that required detection limit goes lower and lower. So it increases the required sensitivity or lowers the threshold for detection. And again, lowering the threshold means that that's bad. That doesn't mean it makes it easier. Quite the opposite. It makes it harder for that analytical scientist to meet. So you have a much more higher burden of sensitivity that you have to meet the longer the lifetime exposure you have to the particular medical device. Now, I want to dive into some of these core analytical chemistry techniques to help us as engineers and fledgling medical device developers understand how to apply these for chemical characterization and later a toxicological risk assessment. So, all analytical methods have their own analytical threshold below which they're incapable of detecting compounds. And that's very often and very much governed by how much noise, how much variability that they have. And these things can be optimized through calibration, through tuning, through adequate cleaning and maintenance of the equipment. And the more you neglect those things, the more noise and variability you're going to get. So, very typical sort of chromatography output would be something like this graph, where you get this really kind of wavy noise peak, and then you actually get your analyte coming through, which pops up some sort of blip on the radar, so to speak, and that represents your analyte. And the difference in that height in that area that you have between your true signal and your noise signal represents your signal to noise ratio. And from that signal to noise ratio, there's two things that we really want to describe with respect that's going to come into play to our analytical threshold. And that's both the limit of detection and the limit of quantification. Now, in layman's terms, these sound somewhat synonymous, but they have strict analytical definitions. The limit of detection, or LOD, is the analytical limit where we can reliably detect, as in say that something is there, and it's defined as having a signal to noise ratio of 3 to 1. There's actually some standards that describe other ways of describing these, but this is a great way for describing with respect to mass spectrometry and chromatography methods. The other accompanying definition is going to be the limit of quantification, or the LOQ. And that's the analytical limit where an instrument can not only detect, so not just detect, but also quantify the analyte. So one sort of a qualifying metric, and the other is a quantifying metric. And in order to understand the toxicological concern, we're going to be concerned with the LOQ, the quantification limit. And that's defined as a signal to noise ratio of 10 to 1. So if you can imagine, noise is noise. You can't get around that. The LOQ has a much higher burden in that your peaks need to be at a 10 to 1 ratio of your noise limit. If they're below that, you're essentially saying, I can't reliably say what amount of stuff is below that, and I generally have to assume it's zero. So my analytical sensitivity limit is going to be dictated by whatever is 10 times what my noise ratio is going to be. And once we get our hands on some actual data, it's going to be a lot more apparent how we actually quantify that. But at least hopefully visually, looking at just some of these chromatography graphs, we kind of get a high level exposure of what that actually means. Now, I just sort of described two broad thresholds that I want to recap and go back over these because they sound maybe not similar, but I very often got these confused early on in my learning history of this. So, again, the analytical evaluation threshold is something that you determine upfront before you start any experimentation. It's going to be governed by a lot of your experimentation methods. But as we saw, it takes into account the DBT, these kind of rules of thumbs that are already predetermined, and also based on some factors on our experimental methods, what our dilution ratio is, how many medical devices we're actually exposing to these E&L testings, the volume of our solvent extraction, and so on. So once we decide on our protocol, we know exactly what our AET is. The limit of quantification, on the other hand, is another core thing that we need to determine, but it can only be determined after experimentation has been done and is qualified through experimentation. So, based again on that signal to noise ratio, it's going to be able to tell us what we're capable of seeing. And it has nothing to do with toxicology at all. It's a pure analytical description of our sensitivity and capabilities. So, AET is what the FDA is concerned with you being able to detect. Because if you can't detect anything below that, then how can you even say that something risky isn't there? You're not going to be able to detect it. And it's not because it's not there. It could be for all the FDA cares, and they're going to take the worst case assumption. They're going to assume it's there unless you can prove otherwise. The limit of detection is simply what you're able to detect in the first place. So I think we're already starting to see the harmony between these. What the FDA wants to see, what they mandate in order to be confident that nothing is below that AET, is that your limit of quantification is positively below that analytical evaluation threshold. If you can't have an LOQ that's below your AET, how can you possibly confidently say that there's no risky materials at the AET or below or above concentration? You've already acknowledged that your LOQ, if it's above it, that there's some sort of gap or window where you can't say anything. So that's why this is a hard requirement for any chemical characterization and toxicological risk assessment. No, yeah, hard stop. So we've covered the extraction and the detection steps. Now we want to move on to the identification steps. So I don't want to go too much into detail about the actual methods unless we're actually running them, but I did want to cover mass spectrometry just a little bit, just because we're going to be working with this and we're going to be trying to understand the data somewhat. So in general, chromatography is the process of simple separation. It has nothing to do with identification in itself, but it separates things out based on their polarity, like with liquid chromatography, or maybe volatility for gas chromatography. And there's a lot of different ways we can go through and describe that chromatography method. But in general, they're just going to separate out some things. The mass spectrometry part is what actually helps us with the identification process. And that's where compounds are ionized and then fragmented to look at their unique mass spectra. And by understanding their masses and where and when they come off, we can use that to identify what those compounds are based both on their separation profile and their masses. Now, whenever we try to look up their masses and their separation profile, we'll look at some sort of library matching technologies to see how they compare with other things in the past in order to make that targeted comparison. So we can actually do that through a direct targeting method where we're not looking at any libraries. We have known reference standards. And oh, if I want to look for polyethylene glycol, well, then I test for polyethylene glycol at that mass. And I have known samples that I put that on a mass spectrometer. And then I look at my extraction solution. And do I have something at that separation point and that mass that also comes off? Then I know that because I targeted that compound, I'm going to be able to see that directly. I can also do sort of an indirect matching where I look at a library profile or history, and there's quite extensive ones out there, where I can do sort of a broad screening method and look for many different things against extensive libraries and reference spectra and help me identify rapidly all the things that came off. So there might be cases where you have a direct concern with something and therefore you want to target that particular compound. Or in a broad screening, this is my first time looking at the chemical profile, I'll do a broad, non targeted method to see what's out there. Now, in both cases, I need to know exactly how much of a thing that comes off. And if you look at some of these spectra that's on the right, you can kind of see I'm not getting like a true concentration thing. I'm getting some intensity, some absorbance profiles. There's a lot of different ways to detect something, but none of them are going to be directly related to concentration. So therefore, I have to compare these to different concentrations and somehow infer and calculate the concentration profile of known or unknown surrogates in order to do that. I'll talk more about that stuff, but you kind of do that in two different categories. You either use those targeted standards where I know exactly what I want to look at. And I'm just like in that polyethylene glycol example, I'm going to have different concentrations of PEG. And then I'm going to see where my unknown extraction possible PEG compound comes off of what intensity and look to see where it lines up with those different concentrations in order to interpolate where it is. In the case where I'm doing a non-targeted method, I'm going to have to use non-targeted surrogate standards where I'm acknowledging, hey, I can't detect everything in this, 5,000 reference library spectra compilation that I'm going to use. So I'm going to test a variety of different compounds of different polarities, different solubilities to try and cover the gamut of different organic molecules that I might look at. And then whatever reference or whatever compound that I find, I need to somehow associate with one of those surrogate standards. You're never going to use just one surrogate standards because you're doing such a broad sort of screening method. You have to use a variety of surrogates and then somehow match the closest surrogate based on chemical composition or behavior, polarity, what have you, in order to match up with your unknown. And I'll describe exactly how we do that on this next slide. And we do that, we use those standards in order to come up with concentration curves or standards curves. And this is where I'm going to take a known compound, whether it's a targeted or a non-targeted surrogate, and I'm going to make a controlled exact concentration. Because I'm making in the lab, I'm going to have full confidence in what exactly it is. So say I start off with a 1 milligram per milliliter concentration. Then I'm going to go through some sort of dilution factor, a serial dilution step where I make it a different and higher dilution ratios. So then I can have a variety of samples that are all at different concentrations. Then, if I test that on my mass spectra, on my chromatography method, I'll have known concentrations or samples of known concentrations going through, and I can correlate that to whatever intensity or absorbance profile, whatever detection method I'm using, that's going to show up on the y-axis. So I have my known x-axis concentrations, and I'm going to see where they fall on that y-axis. Again, from there, I can say, oh, if I put on blank samples, or I can see where my noise is, what my variability in those samples are. And then I can make a basic y equals mx plus b line if it's a linear concentration dependence with whatever my detection method. And that's going to tell me my sensitivity, my range over which I can detect these things, both my upper limit of detection and then my lower limit of detection and quantification. Again, based on those signals to noise ratios. So that's what the process of getting a standards curve. And then whenever I put on my sample of unknown concentration from my E and L procedure, I'm going to get a certain signal from that, a known Y, and I back calculate to see where it falls on that Y equals mx plus b line in order to get my X concentration from that unknown sample. Now, from that, a typical output report is going to have a method section, a standard section that talks about your limit of detections, that talks about your slopes. But whenever we get into the chemical characterization results, what actually came off from your E&L testing, that's where you might have, well, here's the sample that came off my gas chromatography mass spec from my hexane extract. And here's a table of a compound where I found polyethylene glycolacetic acid that came off my liquid chromatography method from my isopropyl alcohol extract. And it kind of gives you, well, here's the molecular weight. The retention time is actually when it came off of my chromatography method. And then based on my uh, I converted my concentrations to my actual dose that a patient would receive in terms of microgram of this particular toxin per gram of medical device. And then you see here, I even listed the surrogate standard that was used to achieve the concentration. And somehow there's some argument or justification on how this particular surrogate best applied to that particular standard. So this would be one of the final outputs of your chemical characterization report in order to understand how much of a material came off per mass of your particular medical device. Now, as I mentioned before, it's pretty rare that you're just going to do chemical characterization. Maybe if you're just doing pure equivalency testing, hey, I've already done this before. I changed my raw material. I just want to show how similar it is. Maybe you don't need to do the next step, which is the focus of here. But in general, you're going to inevitably, certainly for the first time, have to accompany chemical characterization with that so what assessment. And that so what is a toxicological risk assessment. No one cares in a vacuum how much of a particular material is present or gets extracted from a material. We only care about the toxicological risk that accompanies that particular material. So now we're going to shift to the -17 ISO standard that governs and discusses the TRA, or the Toxicological Risk Assessment. So the definition of the toxicological risk assessment is that it's a method for determining the allowable limits of substances that are leached from your specific medical device. So this isn't to be confused with the AET that I discussed previously. The AET and the toxicological threshold of concern are simple like rules of thumb, are guidances for the initial screening process to help guide the analytical methods and use to describe the sensitivity that might be required to identify all things of general concern. So that those get applied before an actual test takes place. The TRA is put in place after an analyte has been identified and is specific to that analyte itself. So AET and TTC are just broad things. They're not specific for any particular toxin. They're just general rules of thumbs that toxicologists have come up with. The TRA is specifically to applied to each and every analyte that was found in chemical characterization. So, the standard defines safety of a medical device is that it requires an absence of unacceptable health risks. So, what we're going to do is go through each and every analyte in order to understand what is the risk of that analyte and is that acceptable. Your medical device will only be deemed acceptable and shown to be reasonably safe if you can show an absence of any particular health risk with any analyte that you show. Now, there's two core components of a toxicological risk assessment done on a per analyte basis. The first one is determined entirely from toxicology, literature, prior study derived values. And that's determining what we call the tolerable intake. And that's where we estimate the average daily intake of a substance over a specified time period on the basis of body mass that is considered to be without appreciable harm to health. And this could be considered the equivalent of the toxicological threshold of concern, except this time is specific for the unique analyte. So if you found that you got exposed, that polyethylene glycol comes off your device, you look into and you understand, okay, what if toxicologists determined to be an unreasonable level of that particular compound, convert that into a dose that that particular, person would receive, and you kind of understand here's the sort of tolerable intake, the maximum allowable amount of this, where then after which I would start to get harm. Then you look at a result that you get exactly from chemical characterization, the quantity of your material, and you convert that raw extracted concentration. That's typically in the order of, microgram of analyte per the amount of your medical device. And you want to convert that into a more practical, not I have this medical device in a jar and this is what came off of it, but okay, how much of this medical device would I be exposed to? Is it a short term, a long term contacting thing? How many medical devices or equivalent would I have in the body? And let me convert that concentration into a human dose equivalent, not in terms of grams of device, but in terms of per person intake for an expected actual exposure of that device. Now you would compare those two numbers, basically comparing how much of an identified analyte can a person tolerate hypothetically, and then how does the number that I actually recorded compare to that? And that's at that point where you might look into thinking how you might be able to reduce that number, or even if you somehow get exposed to more than is applied of a toxicological concern or is governed by the tolerable intake, you don't necessarily fail right off the bat. That's where you can justify to a regulatory body, well, the benefits of my medical device are so beneficial that they outweigh any, slight amount above this toxicological concern or the more life and death impact that your medical device might present, the more you can justify having exceeded some sort of tolerable intake. So that's what I mean by regulatory justification. Now, sort of understand some of these terms, I want to go a little bit of a toxicology 101 and talk about how we actually assess what a toxicological concern might be for any particular analyte. And this is where we want to get a dose response relationship out of any particular adverse symptom or event associated with a particular analyte or device. So, what you see here is a typical graph where you first want to define what your adverse response would be. It could be in a cell culture study, cell death, the cell mutagenicity, if it's potentially cancerous, its ability to prevent differentiation in stem cells, which might relate more to the reproductive toxicity of a device. It could be in an animal study, the rate of disease development or even death of animals. And then in humans, the same thing. It could be the presence of certain symptoms, fevers, the development of certain diseases, or potentially even death. Now, toxicologists wouldn't want to evaluate that, of course, in humans directly, as in doing an active study where they look at that. But it's in cases where people have been retrospectively exposed due to some sort of chemical leakage, uh, being exposed to a particular chemical weapon, for instance, where we might be able to get this type of data from humans directly. Otherwise, it's typically cell or animal related studies. Then we will look at the dose that was applied to those particular subjects, and then kind of understand and plot that dose response threshold. And there's two key terms that we want to look at here. There's what's called the no observable adverse event level, the NOEL, which is the highest dose that showed no response. And then the accompanying term is the lowest observable adverse event level, which is the lowest dose that showed a positive response. So one is the highest dose that failed to show a response, and one is the lowest dose that recorded a response. And typically, because the NOAEL is the more stringent of the two, the lower concentration, that's what toxicologists will want to look at. And very often, there might be a big gap between the two just based on how the study was carried out and due to not wanting to incur unnecessary animal deaths or particularly human deaths, we might not have that big gap. So we have to rely on that lower concentration until we had a more complete picture, which is why the NOAEL is typically chosen. Now, going back to our toxicological risk assessment, we would then want to use a number like that to calculate the tolerable intake calculation, which again is not based on the experimental methods other than the presence of an analyte. It's simply based on literature values that we come up with in order to assess the tolerable intake. So you would take that more stringent of the two, the NOELs, as it's more preferred over the low observable adverse event level, and that would form the numerator of your tolerable intake. And then in order to assess its applicability to your particular situation or to humans in this case, we then need to apply certain uncertainty factors based upon how that study was originally carried out. So for instance, uncertainty factor 1 typically accounts for inter-individual variations among humans. So this is just a default value. And by the way, the ISO standard lays all of these typical numbers out. You don't need to know them off the top of your head, but just the basis that people are different and no one study can account for all these variances in people, a tolerable intake is going to be inflated by a factor of 10 just based on the fact that we can't understand these variations. Now, uncertainty factor number two is going to account for the extrapolation derived from species other than humans. So, if you do a primate study, a goat study, any other type of animal study, that would certainly give you some insights on the toxicology of something, but all species are different. So, just the fact that you're trying to um, interpolate data on a different species and apply it to some sort of uncertainty in humans, you applied uncertainty factor of 10 just based on that species difference. Now, uncertainty factor number three will account for the quality and relevance of the experimental data. And this really takes a number of professional judgments, but a few considerations the ISO standard lays out is that short term studies over a few weeks, maybe even months, often have to be used to extrapolate and understand long-term exposure. And when we need to do that for a lifetime implantable medical device, we have to apply an uncertainty factor of anywhere, from 1 to 10 just based on that. If you only have the lowest observable adverse event level from some sort of study instead of a no observable adverse event level, that again is going to result in an uncertainty factor based on the fact that one is more stringent than the other. If you have a study that looked at oral inhalation, whereas your medical device is implanted as a cardiovascular stent right into the blood supply, then that's going to have a different route of exposure and again require an uncertainty factor modification. And then there's varying rates of exposure. Maybe in an animal study, animals were dosed once a week, whereas in your particular medical device, you might have constant contact and duration. So that's going to require its own uncertainty factor. So there's a number of different things that can modify this uncertainty factor number three, and the ISO standard goes a little bit more detail about it, help you pick out your exact numbers for those. Now, furthermore, the ISO standard even gives a pretty handy sort of flow chart to help you understand how to exactly compute these things, understand what the quality of your data is, and how to go about calculating these tolerable intakes and human dose equivalent exposures in order to do this type of test, whether or not it requires further modifications, if it's a carcinogenic compound or not. This flow chart is pretty handy in helping you go through an analyte by analyte sort of evaluation. Now, the human dose equivalent, on the other hand, is based entirely on the actual results that you got and the quantifying concentrations that you got in your chemical characterization method. So it's simply the human dose equivalent as a product of your ChemCare analyte in terms of the micrograms of your analyte per gram of implant, multiplied by the exposure that implant to a person per day. So a lifetime implantable, they would be exposed continuously on a daily basis. You might have more than one medical device. You might have tested a different extraction of that medical device, for instance, half of the medical device. So you might need to actually put in the full implant size in order to calculate the HDE. And then you need to incorporate the size of the person that you're testing this for. So the ISO standard gives standard values for adult men and women. You might have medical device geared towards children or even neonates, where their size amounts will be even different. So those will give you different qualifying factors in order to calculate your HDE. Finally, you put these numbers together in order to understand your margin of safety, which is simply the ratio of the tolerable intake to your exposure dose, your human exposure equivalent. Now, really, what the FDA wants to see with most regulatory bodies is that you have a margin of safety that's greater than 10. Now, margin of safety is greater than 10 are what most regulatory bodies are looking for. When you're greater than that, you can confidently say that exposure to that analyte can be considered to not be a risk to the health of the patient. And that's because the margin of safeties are shown to be conservative in themselves, and even that ratio of having a margin of safety of 10 is in itself pretty conservative. Now, when you're between 1 and 10, that means you're still over the tolerable intake, so you're still under the tolerable intake, excuse me. But you still need some sort of justification on the exposure that comes into play with the benefit of your particular device. And so whenever you have a higher exposure of that analyte, compare the tolerable intake, that you don't necessarily fail all of chemical characterization, but the FDA is going to look pretty stringently on any kind of justification. You're not just simply showing you're close to that margin, to being close. You've exceeded it, and you have to have some sort of justification or a standing benefit that far exceeds the risk exposure that you're acknowledging from that toxicological concern. Now, if we want to understand how this kind of look pulls together, I posted a sample outline of just one of my prior tests in chemical characterization and the toxicological risk assessment. These are typically reported in paragraph form, where it showed that, hey, chemical number one was identified by this method. In this study, where an acute oral toxicity was conducted by Smith et al., these rats were fed this chemical orally and was found this, da, 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 da. And that study determined that the no observable adverse event level was this. And here's another study where they found, a different route of exposure, a different slight, study outline, and they got a different no observable adverse event level. So it even plays upon multiple studies, has multiple no observable adverse event levels. You can take the most stringent of those two, you say selecting the more conservative of these, and applying this modifying factor of 1,000, which is based on a factor of 10, 10, and 10. Since the second dose was a bolus injection, this was a long term, you come up with a tolerable intake of that particular amount. And then you can compare that tolerable intake to the human dose equivalent and have a margin of safety. And you typically see these in a big tabular form, a big table at the end that is rooted or based upon all these sort of paragraph form, analyte by analyte descriptions. So that's going to be the typical end result of your toxicological risk exposure. So hopefully that puts in some insights on the full range of testing from chemical characterization to TRA and the full outputting of the results and what's needed in order to demonstrate part of the biocompatibility of your device and then potentially justify based on what you found here if further studies are needed or warranted based on your device and recommendations from regulatory bodies. Thanks for listening.